Controlling gene expression. Another strategy is blocking kinase synthesis. Kinase is a protein. Then to make a protein, we need mRNA. And to make mRNA, we need the DNA. So instead of attacking directly kinase, if you attack mRNA or DNA, then eventually they cannot make this protein. So this is another strategy to abolish the kinase activity. To attack the mRNA, one approach is utilizing short nucleotide, which has a complementary sequence to the mRNA. That complementary sequence, in a sense, that is called as antisense, because mRNA is a sense strand. Once they form this kind of double helical RNA structure, it can be target of RNase H1, it can be degraded. So mRNA is removed. That's why target protein cannot be made. So this is very interesting approach to abolish the synthesis of a target protein. The problem is this antisense oligonucleotide, either DNA or RNA, these molecules are connected by phosphate bond, which has a high D negatively charged. So that's why these molecules has a problem for the cell penetration. So delivery to the target site is the difficulties of these molecules. That's why this approach has been limited only to the eye drug, the drugs, which has an open the target to the access of the drug molecules. Instead of a single-strand antisense oligonucleotide, if similar size of a double-strand RNA is added, our body recognizes this as potentially invading virus genome and then dissociate those double strand into single strand to suppress those virus functions. Interestingly, if this sequence is matching to existing mRNA, this single strand defragment can recognize mRNA and then cleave off this mRNA. So this discovery is called as siRNA. If you add double-strand RNA, then it can cleave the mRNA much more efficiently than antisense oligonucleotide. Maybe the high efficiency come from regenerate of this double-strand siRNA, and then it's amplified the signal to break down the mRNAs. Similar size of a double-strand RNA is also discovered with the name of microRNA. While siRNA bind to the coding area of mRNA and destroy it, and usually siRNA has a perfect match, microRNA, it binds to either 5' or 3' area, and 3' also has a poly A tail for protection. Now, microRNA, in many cases, they have a 1 or 2 base at a mismatch, so it's a loosely bind to this controlling area, and in many cases they suppress the gene expression, but sometimes it can also activate the gene expression too. So siRNA is usually synthetic molecule we make in the laboratory and add to the biological system, but microRNA seems like these molecules are intrinsic natural regulators for gene expressions. CRISPR is a currently very famous gene editing tool, and this was discovered from bacterial gene sequencing. In bacteria, there is repeating genes, and then in between, very interestingly, the virus fragment gene information is inserted. So scientists suspected this may be the bacteria's defense system against the viral infection. Usually, if a virus attacks bacteria, virus wins. But if bacteria wins the infection, and then they wipe out the virus, and then save small amount of the viral gene in their genome. If the virus attack again later, utilizing this fingerprint, they develop the defense system, they cleave out those genes. So this is the discovery of a CRISPR system. Let's look at the mechanism of CRISPR. If that bacterial gene is transcribed, then it makes CRISPR RNA, and it contains viral information here. If this matches to the invading virus's genome DNA, it binds to it. And this repeating unit, this can bind to trace RNA, and this complex can recruit enzymes so-called Cas9. If Cas9 binds to this complex, it can cleave the host gene's DNA. The original CRISPR system, they required 2 strands of RNA, but to make it easy to use, it can be connected together and then to make single guide RNA. Originally, CRISPR system bacteria used to attack invading the viral genes. But it was very soon known that this technique can be used in any type of cells. Then, if you design this sequence for the matching to your target gene in any type of cell, then just by introducing Cas9 together, now you have a very powerful tool to attack and cleave your target genes. A modified CRISPR system, CRISPR-A, is also possible. Original function of Cas9 is binding to the sequence and cleave. But if you mutate the Cas9 to make it deactivate the Cas9, it can bind to the sequence, but it cannot cleave. Instead, here if you add transcription activator, and then this binding site is not in the coding area, this is a promoter area, then this binding event can accelerate, turn on the gene expression. So this is the activation. That's why this is called as CRISPR-A system. Very similar way, it can be also used for inhibition, CRISPR-I. Instead of activator, if you add a suppressor gene here, then it can block the synthesis of gene expression. Let's compare those 3 techniques. Original CRISPR is by binding to the target gene, and Cas9 cleave the target gene. So this is deletion of the gene. That's why this is called CRISPR knockout. CRISPR-I, it does not affect the DNA itself, but that blocks the synthesis of a new mRNA. So this is a knockdown experiment. And this is very similar to siRNA, but difference is this is blocking the synthesis of mRNA. siRNA is destroy the mRNA. CRISPR-A is opposite. That can accelerate the transcription of that mRNA. So if this is a knockdown, this can be maybe knock up. We don't call it that way, but in that sense, it can be called knockdown and knock up.